Hi everyone, my name is Kevin Labadie, and I'll be joining you on your journey to Step 1 by reviewing the remainder of the microbiology chapter. First, I want to thank you for choosing First Aid Express to aid in your Step 1 studying. I promise you that our entire team is working very hard to make these videos informative, fun, and worthwhile for you. As we're watching these videos together, there are two things that I ask of you. Most importantly, I want you to be engaged in these videos. I'll be asking questions throughout the videos, and I want you to try writing, drawing, vocalizing the answers. By engaging different parts of your brain, such as these, you'll be bringing this material deeper and deeper into your memory. Second, I want you to provide feedback to me when you can. Learning and teaching is a dynamic process. I'm always looking for ways to improve and make this material easier for students. So now let's start by talking about virology. From my experience with teaching medical students, virology tends to be disliked by many. It involves a lot of brute memorization, confusing terminology, and seemingly arbitrary clinical application. My goal over the next several hours is to cover this material in an organized way to make it easy as possible to digest and store in your mind. So let us start by discussing the basic properties and structure of a virus. With a solid understanding of the basic structure and organization, we can begin to categorize these viruses into classes and groups. To do well on this section on your Step 1 exam, it is important to have a solid understanding of how these viruses are classified. So let's begin. Alright guys, so first, what is a virus? Have you guys ever thought about that? Well, a virus is an infectious agent that replicates only inside other cells. This virus requires these cells because it does not have the machinery needed to replicate and become infectious on its own. It relies entirely on other cells, including cells of our human body. Think about it like a car factory. To make a car, there needs to be machines and tools and people to make the car. Without the appropriate factory or people or tools, there's no way to make the cars. Same concept here with viruses. The virus needs to infect human cells or animal cells in order to access the machinery and the tools to make more of it and continue to propagate its genome. In general, viral structure has three important components. All viruses have a genetic component, we can see it here, that is composed of nucleic acids, DNA or RNA. It also has a protein shell, known as the protein capsid, and finally it has the lipid envelope, kind of seen here. When these are all put together, it is known as the virion particle. Unfortunately, just to make our life confusing, as biology often does, not all viruses have these lipid envelopes. But don't worry, we're going to spend a lot of time going over which ones do and which ones don't in the remainder of the lectures. The protein capsid can take one of two shapes. The key shapes it can be are icosahedral or helical. Icosahedral is like a polygon structure here, a pentagon structure, while helical is like a helix, like that. The protein capsid has a number of important functions, but I want to spend some time focusing on two. First, the protein capsid acts as a protective shell. It holds the viral genome, the DNA and RNA, as well as the viral encoded enzymes into one little sphere. After the DNA and RNA has been synthesized in the host cell, once it's been made by using the host cell DNA machinery, the molecules, the DNA and RNA and enzymes are packaged into the protein capsid. Second, capsid proteins serve as receptor ligands, or receptor proteins that initiate uptake and infection into victim cells by binding receptors on those cells. The proteins on the protein capsid are molecular keys that bind to the victim cells and initiate uptake. You can think of these receptors or ligands, protein capsid ligands, as hooks that as they travel around the bloodstream, they look for the right hooks to bind to and then allows for the virus to infect the tissues. Without these proteins anchored into the protein capsid, the virus is not able to infect or initiate infection into any cell in the body. These same capsid ligand proteins, these same proteins that are responsible for binding receptors on human cells, are also what is recognized by our immune system. Or this is the antigenic stimulus that allows for an immune response. Now thinking back to our immunology, do you remember which immune cell typically responds to viral infections? That would be our good friend the T cell. Thinking back to our biochemistry real quickly, do you remember what a ligand is? What is a ligand? A ligand is the functional part of a protein or molecule that binds to another protein or molecule to form a coordination complex. Put simply, the ligand is a part of a molecule or protein that binds to another one, such as a receptor. If we think back to pharmacology, to extend this example further, drugs are ligands, just like viral and bacterial proteins. They bind to receptors and cause something to happen within the cell. Now that we've discussed the protein capsid, let's talk a little bit about the lipid envelope. The envelope is the outermost layer of the virin, and it also has two important functions I want to highlight. Number one, the envelope, just kind of like the protein capsid, aids entry of certain viruses into host cells. It works with the protein capsid to bind cells and have the virus infect them. So it's used for viral entry. The lipid envelope also disguises the protein capsid layer from the host immune system. Think of it as Harry Potter's invisibility cloak. You put it on and it hides the virus from the immune system and prevents the immune system from reacting to it. So the lipid envelope helps with viral entry as well as disguising the virus. So now the question is, where do viruses get the lipid envelopes? Does it synthesize it like the protein capsid? Well, not exactly. The viruses acquire their envelope from the plasma membrane of the host cell as it exits. As the compiled virus begins to leave the cell after its DNA and RNA and proteins have all been synthesized, it pushes through the plasma membrane and kind of bites off a piece of it and takes it with it. Remember, this is from the plasma membrane of the host cell. But, as with a lot of medicine and biology, there are a lot of exceptions. And do we remember the one exception to the rule is? I know we haven't covered this yet, but the herpes virus family obtains their envelope from the nuclear membrane. So now that we have these three viral components in mind, what are they again? Nucleic acids, DNA, RNA, protein capsid, and lipid envelope. Let's begin to organize and categorize these viruses. I cannot emphasize how important categorization of viruses is for step one. This is the material that's going to be tested. First, the viruses can either be DNA or RNA viruses. The nucleic acids make up the genome of the virus. Think about the genome as the blueprint of the virus and is very important to the ultimate structure of the virus. These nucleic acids, or this genome, is packaged, remember, into the capsid protein, and some have lipid envelopes. This is what the virin is, just to review real quickly. Each virus can take one of three different forms. The virus can either be naked or have an envelope. It can be, as I mentioned earlier, icosahedral or helical in terms of its protein capsid. So now we're going to combine these and show you the three types of forms that a virus can take. So first, a virus can be naked with an icosahedral capsid. When I say that there's a naked virus, that means that there's no envelope. So number one, the virus can be naked and have an icosahedral protein capsid. Number two, a virus can be enveloped with an icosahedral capsid. So here we have a naked virus with icosahedral capsid, and here is an enveloped virus with an icosahedral capsid. Very good. And finally, number three, we can have an enveloped virus with a helical protein capsid. So what we notice here is that all helical capsids are enveloped. There is no naked helical capsid viruses. This is a very important point. There can be naked and enveloped icosahedral protein capsid viruses, but there are no non-enveloped helical capsid viruses. All helical capsid viruses are enveloped. Very important point. The way I like to think about this is that a helical capsid protein structure is not strong enough to protect the virus without an envelope, while the heavy-duty icosahedral protein capsid is sufficient for protection. We can think of the icosahedral structure as a thick trench coat, while the helical capsid is more of a long-sleeve t-shirt that needs a lipid windbreaker or coat to keep warm, for instance. So the icosahedral protein capsid is stronger than the helical protein capsid and therefore can be naked. Helical capsids can never be naked. They always have envelopes. This is very important.